In this module, we will further continue the different types of cell transduction, different molecules which take part in the cell transduction during the process. Now, the cell transduction or signal transduction components, number one is the receptor. Now, what are receptors? Receptors are, as a matter of fact, they bind on the cell surface to their ligands. Ligand, mind you, are chemical compounds, chemical components, or somewhat related to them. Now, signal molecules, they are known as ligands, and they are bound to the receptor via their extracellular domains, because now the cell, it develops the receptor on the surface of the cells, and binding specificity and affinity are determined. They are very important, but they are determined by the extent of molecular complementarity between the ligand and the receptor. Molecular complementary mean, just as if you remember, in the enzyme we have lock and key model where those things fit. Similarly, the signal, it has to fit to the affinity or to the complementary between the ligand and the receptor molecule, which is developed on the outside of the cell. For example, here, that is, there are essential component, that's a growth hormone, which is there. Then it is giving the signals, and it binds with the residues which are essential to the right binding with the hormone. And there are amino groups, there is hydroxyl group at the bottom, and it joins together, and further it makes a complete module where there it fits into the growth hormone and makes the signaling possible. Now, proteins that participate in intracellular signal transduction, they fall into two domains, into the two classes. One is the protein kinase, which is the phosphatases, and GTPase, that is the switch protein. Remember, because now there are signal transduction and there are electrical impulses, it means that we have to have some proteins which act like switch, on and off, on and off, and that is their function. For example, here you can see that the protein kinase, it becomes active and then inactive. Then protein kinase number 2, it is active and then again inactive. Protein kinase, which is number 3, active and inactive, and eventually the signal is transduced, signal is transmitted to the DNA and virtually to the messenger RNA for the transmission, or to the cell, the signal. Now, as we say that the component for the signal transduction, that is GTPase switches, GTPase switch protein, that plays an important role in intracellular signal transduction. Because wherever there is signaling, it has to be on and off, and that is done by GTPase switch. Now, GTPase, they are active when bound to GTP and inactive when bound to GDP, that is the phosphate group is removed from them. So that is how they become active, how they become inactive. They become inactive when they are bound to GTP, and inactive when the phosphate, inorganic phosphate, is removed from them. Now, GTPase switches again, the time frame of activation, it depends on GTPase activity of these proteins. Now, there ought to be a time frame. It is not that a switch is left open continuously forever or for some time. There has to be certain time when it is on and the time when it is off. Signal transduction component, an important role in this area is the second messenger. We discussed earlier the first messenger. Now, the second messenger, they are large number of extracellular molecules, extracellular receptor ligands, and they are called first messenger. But there are relatively few small molecules which are present there, and in the intracellular signal transduction, they are known as the second messenger. The first messenger, they are extracellular, and then intracellular components, they are the second messenger. That is, those components which are present in the cell and which carry out the message which they receive. Now, second messengers are small molecules. They have to be small molecules because they are inside the cell and they diffuse rapidly through the cytoplasm to the protein targets. Remember, in the cell, they are there, small molecules, and they diffuse rapidly. Another advantage of second messenger is that they facilitate the amplification. Amplification means enlargement. The signal is enlarged, enhanced, and that is of the signal which we receive or which the cell receive from extracellular messengers. For example, the second messenger, they are, because they are ligand, so they will be chemicals like cyclic AMP, then the cyclic GMP, they are activated, and further there are other areas of those molecules, other molecules which accumulate, which transduce the signals to the cell. Signal amplification pathways, there are two types of pathways. One is the participant in the pathway increases. This is referred to as the signal amplification because in that pathway the signal increases. This is referred to as the signal amplification, as I said in the earlier. And for that, what it has to be? It has to be maybe hormone, maybe signal pathway, often referred to as amplification cascades, that they amplify from one stage to second, third, fourth, fifth, and so on. For example, here we take another compound that is epinephrine. That is 10 raised to power minus 10 molar, its concentration. And we can see that it passes on to the one state that goes to the adenyl cyclase, then cyclone AMP, and there is amplification. You see, after that, the one signal, it goes to the fourth signal. Then the protein kinases, which further activate the enzymes and amplification. And at the end, there is the product which is needed for that process, and through the amplification is there. Now, example of signaling amplification, that is one epinephrine activated GPCR activate hundreds of GPTAs to GTP complexes. Hundreds, because there is amplification, which in turn again activates 100 of the adenyl cyclase, as we saw in the previous diagram. And there are molecules, the cyclase molecules, and each product, what it does, there are hundreds of cyclic AMP molecules, and so on. Therefore, the overall amplification, सब जो उसमें increase है signal में, वो क्या होगा? That is associated with epinephrine and signaling, and that will be about 10 raised to power 8 times fold, 8 times more. अब आप देख लें कि ये कितना ज़्यादा उसका सिग्नल एम्पलीफाई होगा थ्रू द कैस्केड एक्शन.